Hello everyone. The topic I want to introduce today is the histological finding of sperm storage in green turtle oviduct. And we all know that the green turtle is on the red list of endangered, like eating too much plastic bag or has the straw on his nose. There are so many reasons make them as endangered. And this is important to understand the reproductive organism of green turtle because it can help us to figure out more efficient way to conserve them. And the histological means the structural science. And the green turtle is a seasonal breeder. Each year they will migrate from the Honshu coastline to the Ogasawara Island to mate. And Ogasawara Island is the biggest habitat of the green turtle to mating. And this is on the North Pacific Ocean. And it also where our sample from. And as the paper, you can see in the wild, the green turtle will mate on March to May. And during the 2 to 3 months later, they will lay the egg in early May to the late September. But compared to those turtles who raised by the human, the time days between the mating and the nesting only take about 5 weeks. So we can think that the green turtle has the ability to storage their sperm in the body after the mating. So this is the purpose we want to know. And this figure, you can see the oviduct. The oviduct comprises of 4 parts. And because the turtle's oviduct is too complex, so I use the human oviduct to let you know. The first part is the infundibulum. The infundibulum is like human, is body to receive the yolk to this oviduct. And the second part is magnum. The magnum in the turtle, who is the region, can create the protein and wrap the yolk. And the third part is called isthmus. Isthmus is a transition area between the magnum and the uterus. And he got thinner tube wall. And the fourth last part is called the uterus. The uterus is where we get the eggshell. And this picture is the green turtle's oviduct. You can see this part is vagina, and this part is uterus, and isthmus, magnum, and the infundibulum. And the second page, figure B, is called infundibulum. This is the end of infundibulum, and it connects to the magnum. And you can see there is a thinner tube wall. And the figure C is magnum to the isthmus and the uterus is this part. And the magnum got thicker tube wall. But between the uterus and magnum, the isthmus, the tube is thinner, and it stands about 5 cm. And the last figure is uterus and vagina. You can see the uterus, his wall is many large folds, and this symbol means the transition area between the vagina and the uterus. And we got 3 samples from the Ogasawara Island, 3 of female and 1 of male turtles. Epithelium is this red part to see the sperm's condition. And their body length, body weight, and their weight, and their temperature status is shown on the table. And we got a sample from 5 different parts of the oviduct of ID 035. And we got 4 from the infundibulum, 17 from the magnum, and 2 from the isthmus, 3 from the uterus, and 3 from the uterus and vaginal transition area. And likewise, the 024 and 028, we got 4 different parts of the sample. Is infundibulum, magnum, isthmus, and the uterus, but no uterus and vagina. Before we slice the sample and dehydrated the sample, we use the scanning electron microscopy to take a picture. And his mode is here. And like the picture on the right, this is the sperm obtained from the male turtle's epidermis, like left of the page, epidermis. And this is the sperm. This pink area is the sperm's head, and this is the sperm's tail. And before we check out the result, we should know some name of this structure first. And imagine this is a tube of the oviduct, and the center of the tube is called lumen, and the gray part is our mainly sample from. It's called epithelial tissue. In Chinese, it's called 上皮. And in the epithelial tissue, there are many glands and gland duct and gland acini in there. And this is picture we got from the ID 035. This is the epithelium of the isthmus. And this red part is the gland acini, called GA. And you can see the arrow pointing out is a little blue. We use the scanning electron microscope to see it. It's like this. And we check out closer to this white black area. Also this area, you can see there are 5 arrows pointing out. It's all the sperm. And we can know from the ID 024. This is the epithelium. And in the epithelium, this is the gland duct, and the arrow pointing out under the gland duct is some sperm. Let's look at it closer. You can see this insert figure has a little blue line. It's all the sperm. And the figure B is from the ID 028. Sorry, my age is gone. And this individual is captured when she is mating. So in the isthmus, the sperm didn't get into the epithelium, but on the surface of epithelium. And the arrow point out is plenty of sperm. Oh, this area is called the lumen. And the figure C, you can see it's got from the ID 035. And this is from the vagina and the uterus transition area. And you can see this gland duct have a little arrow point out some sperm. So we take it as a table. With double plus in all turtles, their isthmus have plenty of the sperm in the gland. If the dash, it means there are no sperm in the gland. And if only 1 plus, it means there are some of sperm. The ND means they have sperm, but they are not in the gland, but in the lumen or epithelial surface. So the result is we got all sample has the sperm in the gland or isthmus. And we have some in the vagina. We have 1 individual in the vagina and the uterus transition. So we think that is the main sperm storage area, and this may be the other side of the sperm storage. So this question is, does the degradation of sperm when the sperm on the unsuitable area, and how long the sperm can be stored, and is the sperm only attached to a ciliated cell like a soft shell turtle? In case to do this, question 1 and question 3, we need to use the transmission electron microscope to do the further research. And for the question 2, maybe we should take the non-mating season sample. And thank you for your listening.
I want to explain the turtle to storage their sperm.
You want to know why the turtle storage their sperm, right? There are so many animals where storage their sperm because storage sperm can help them to choose the better sperm, and some species like turtle, the male and the female will mate maybe many years. So this is why the turtles store their sperm.
Since the sea turtle is a distinct species, and how do you get the sample?
Our sample is...
Repeat his question.
You want to know even the turtle is retrieved, but how can I get the sample, right? Our sample is got from the harvest and harvest as food, but we got this dead turtle following the fishery adjustment regulation of the Tokyo. So it is no illegal issue.
Flora, how do you investigate the green turtle migration?
Green turtle migration. I think you want to know how do I research the green turtle migration, right? I think this is a further study. People have do that before.
Where were the research get this sample?
You want to know where is the fisher get this sample?
Yeah.
We get the sample from the Ogasawara Island, like this picture.
So I like Irin's talk very much. I think she did a very nice, excellent job because when I was reading the paper, I realized that that was something very difficult for me because you see a lot of, these are actually not biological terms, these are anatomical terms, 解剖学的. So these are not something you hear about in your other lectures. And I think she she had a very smart strategy because I think many of you learned this in your in your high schools. So she used this as a reference map. So that you can refer these. I don't know. When I was reading a paper, I was like, oh, what's that? But then she was nicely by comparing these to that. We had a better idea, so what the scientists were doing. So I think that was really smart. And another good thing of her talk is she repeatedly referred back to this image. So that, because even after this initial introduction, as I often said, the memory of audience was really short in the talk. So even if after she gave this kind of nice introduction, I already forgot what it was about, maybe 2 slides later. So you see that she repeatedly, so many times, using these figures so that you have a better orientation where she was. That was really nicely done. Okay, and then, let's talk about what she can prove. So I see that this is slides with a lot of images, but I think it would be great if you can add 2 slides with text. The first is, do you have that? Do you have a slide where you tell us the exact purpose with a sentence? I think that's always necessary. Otherwise, it's so easy to get lost what all this is about. So give one sentence, give one slide for that. And the second is here, the discussion, my impression was a little bit like that was future work. It's not really discussion. So discussion is really you discuss what happened. To me, it's a little bit like here and here, where you discuss where they can store the sperms. So I would say instead of this kind of like what we can do in the future is like drawing a big picture, I would say instead of that, you give us a conclusion to sum up, summary, what has happened, what was found in this, with short sentences. That would be great. Okay, and then another thing that was a little bit like a missing link to me is, I realized that you introduced this because in these SEN images or TEN images, you have these difficult words. I don't know where is lumen here. What was really missing in my point of view is, what's the relationship of this to this? Yeah, because we know this, and this is a little bit like what is the scale that you are talking about. So next time, please give us a connection, so it's easier to understand. Yeah, and then I also see that you already, like this one is nicely done. Like you enhance. You know, in the paper, it's very common that people use this abbreviation like a EP. You see here, EPGD. This is fine for paper, but it's actually not good for show presentation like this. And what you did here is good. Like you print out the full name here. And I would encourage you to do this for all the images you did. Because this kind of abbreviation, they are not good for communication. So print out all the images. And if possible, try to enlarge your image whenever possible. Because a large SM image have a greater power than a smaller one. Considering you see an image like that, you will feel a stronger feeling for this instead of this. Yeah, and I was actually not being able, you see there was a lot of sperm. I was really thinking, where is it? Yeah, I couldn't see it, but when I enlarge it here, I see, oh, these are the sperms. So make it large. But overall, I enjoyed your talk very much, so thank you, Irin.